Hello everyone, welcome to this new AtlanChim Pharma webinar about reduction of azides to primary amines. Before I get started, I would like to remind you of a few points. Our last webinar in June was about applications of desmartin periodinane in organic synthesis, and you have here the chemical structure of this reagent. In March, we published a new scientific letter on the hydride reagents with a second part of an overview of their reducing power through recent examples. Like all the others, this new scientific letter is available on open access on our website, just at this address right here. Also, last month in November, we published a post on the LinkedIn in our fortnightly The Minute of Chemistry on the reduction of azide as presented in this slide. We will come back to this example later on in this presentation. But in fact, this very efficient methodology to reduce azides gave us the idea of this webinar. You have on this slide the summary of the different methodologies to reduce azides into amines, which we will discuss through recent examples, mainly from medicinal chemistry and also from the total synthesis of natural products. So, amines are very important compounds in various research fields, including pharmaceuticals, dyes, agrochemicals, and so on. Azide reduction here is one of the simplest and most reliable methods for the preparation of amines. In addition, azide function here is resistant and tolerates a wide range of chemical procedures, so it could be therefore considered as a masked amino group, avoiding in this case a protecting group strategy. So for the reduction of azides to amines, catalytic hydrogenation is superior to the others, both from an economical and ecological point of view, especially in a large-scale synthesis. Palladium zero, supported on the charcoal, is widely used as a catalyst in this reaction. However, problems in chemoselectivity are occasionally encountered when the substrate contains functional groups sensitive under the reaction conditions of hydrogenation, of course. So in in the total synthesis of natural alkaloids containing this type of backbone, this chiral azide was hydrogenated to the corresponding target amine in almost quantitative yield with 10% palladium on carbon under 1 atmosphere of hydrogen in ethyl acetate at room temperature. In a recent work of Professor Anessian on the synthesis of azide bridge pyridoazulene of tropine derivatives, palladium catalyzed hydrogenation of this azide under classical conditions provided the corresponding amine here in excellent yield. It should be pointed out that under these catalytic hydrogenation conditions, this internal epoxide here is stable and no secondary alcohol from hydrogenolysis of the CO bond was detected. Here was subjected to hydrogenolysis to remove the benzyl protecting groups here and here, concomitantly reduce the azide here into the amine. This transformation was conducted in methanol using a Pillman's catalyst. However, the desired compound here was isolated along with some N-methylated byproduct, as you can see here on this slide. So these partial N-methylations have been reported in the literature during catalytic hydrogenation of azides, as we have just seen, as well as during hydrogenolysis of M-benzyl amines, as reported here. This compound here probably results from the formation of formaldehyde on the catalyst surface from methanol by dehydrogenation. Reaction of formaldehyde with amines after hydrogenation of the azides in this case will then give the amines which undergo further hydrogenation to yield the M-methylated derivatives. It is important to note that the methanol used in the previous experiment was distilled over magnesium methoxide and stored under argon. Also, high catalyst loading and long reaction times favor this side reaction. This side reaction is also observed sometimes with ethanol. So, in the formal synthesis of D plus biotin, all attempts to reduce the azido group here of this lactone by catalytic or transfer hydrogenation or using the Staudinger reaction failed. To circumvent this difficulty, the reduction of azido-beta-lactam was successfully achieved by transfer hydrogenation using palladium on carbon and ammonium formate as a hydrogen source in methanol to give the amino beta-lactam here in a very good yield, as you can see here. And in situ generation of molecular hydrogen by addition of triethylserine to palladium on charcoal catalyst results in rapid and efficient reduction of multiple bonds, azides, and nitro groups, as well as benzyl group deprotection under milder neutral conditions. For example, in the total synthesis of sugar framework of a nucleoside antibiotic from D-glucose, this anido sugar here was subjected to 10% palladium on carbon and 10 equivalents of trimethylsilane to reduce the azide group here to this amine here, without affecting the paramethoxybenzyl protecting group, giving this crude amine here, which was directly reacted with benzyl chloroformate. The protected amino derivative was isolated in a excellent yield in this two-step sequence. The same conditions were applied to this protected amino acid containing an azido function right here, giving this amino derivative intermediate, which was then directly converted to this alloc protected amino acid in 64% overall yield. Now the Staudinger reduction is compatible with a large number of functional groups and therefore is one of the most employed procedures for the reduction of azides. So treatment of azides with triphenylphosphine or trimethylphosphine led to the formation of an iminophosphorane intermediate here, following this sequence with a concomitant loss of nitrogen at this stage. In the presence of water, this later intermediate is spontaneously hydrolyzed to a primary amine here and the corresponding phosphine oxide. As we will see, this two-step reaction is mostly performed in a one-pot procedure. The versatility of this method is illustrated in the following examples. Oops. In the development of an efficient, scalable, enantioselective synthesis of an HIV-1 and 3 inhibitor, the reduction of this azide intermediate here was achieved in refluxing THF in the presence of 1.5 equivalent of triphenylphosphine and 10 equivalents of water to give the desired amine in an excellent yield on almost 30-gram scale. In the preparation of cyclopamine derivatives, the same Staudinger conditions were applied to this azide here to provide this amine here in 85% yield. The triphenylphosphine oxide formed during reduction was removed by a silica gel column chromatography in this case. It should be pointed out that due to the presence here of this tetra-substituted double bond and this CBZ protecting group on this nitrogen atom, reduction of the azide here by hydrogenation was, of course, not possible. Also, with certain substrates, iminophosphorane intermediates are rather stable in the presence of water and require acidic conditions for their hydrolysis. For example, the Staudinger reduction by treatment of this heteroaromatic azide here with triphenylphosphine gave this iminophosphorane intermediate, which was then subjected to acid hydrolysis to give the 2-aminopyridine derivative here in excellent yield on almost 2-gram scale. As we have just seen in the previous examples, triphenylphosphine is widely used as a phosphine in Staudinger reduction. However, although trimethylphosphine is more expensive, pyrophoric, more toxic, and has an unpleasant odor, Trimethylphosphine oxide here formed during the reaction is water soluble and therefore easier to remove from a reaction mixture than triphenylphosphine oxide, which is notoriously difficult to get rid of. In addition, trimethylphosphine is a better nucleophile and is used in a Staudinger reaction when triphenylphosphine is not reactive enough. For the reduction of this azide here, with this label smoke protecting group under catalytic hydrogenation, treatment with a 1 molar solution of trimethyl phosphine in THF and water, followed by protection of the resulting free amine here with BOC2O, afforded this protected amine in a 51% overall yield. In this Oops. In this other example, treatment of this azide with a toluene 1 molar solution of trimethylphosphine in a mixture of THF and aqueous sodium hydroxide gave the corresponding amine in excellent yield. In the synthesis of a compound in phase 2 clinical trials for the treatment of migraine, this azide here was converted to this amine in good yield by a sequential treatment with a 1 molar solution of trimethylphosphine in toluene, followed after 2 hours of reaction by addition of water to hydrolyze the iminophosphorane intermediate. To conclude this section, we will look at 2 examples where the reactivity of the iminophosphorane intermediate of the Staudinger reduction is exploited to form macrolactams. One of the most challenging steps of the total synthesis of this complex natural product was the formation of the macrolactam ring here in red. So, slow addition of this azide with here a pentafluophenylester moiety to a refluxing solution of triphenylphosphine in anhydrous toluene afforded the corresponding iminophosphorane, which underwent conversion to iminoether intermediate here via an intramolecular azavitic reaction. This later intermediate here was hydrolyzed, to give the desired target macrolactam in 73% overall yield. From the azide, attempts to carry out this transformation by hydrogenation led to the corresponding dimer. In this other total synthesis of this product here, under similar conditions, with a large excess of trimethylphosphine in a mixture of THF and water, this azide here was directly converted to the macrolactam here in 66% yield via the formation of this amine here, which reacted in situ with this activated ester. With a strong hydride reagents such as LH, chemoselectivity, of course, remains a major drawback. Nevertheless, this reducing agent is used on the simple substrates. For example, treatment of this azide with LAH in ether at 0 degree gave the corresponding amine here in good yield. In the context of the preparation of chiral ligands, reduction of this diazide with an excess of LAH afforded the desired diamine here in excellent yield on 3-gram scale. There are many reports in the literature concerning efficient methodologies to reduce azides by sodium borohydride in the presence of various salts. It should be pointed out that in general, sodium borohydride itself does not reduce azides. In this context, different salts have also been used, such as nickel chloride or cobalt chlorides, as we will see in the following examples. Oops. In the synthesis of bridged nucleic acids, the azido group here at 2' position was reduced to an amine by sodium borohydride in the presence of nickel chloride in methanol over a short period of time to afford the amine here in good yield. We already saw this azide in the Staudinger reduction section. In a second approach, the same authors developed a highly efficient nickel chloride catalyzed sodium borohydride reduction of this azide here. This reaction was complete within 4 hours in the presence of 10 mole% of nickel chloride and 3 equivalents of sodium borohydride in methanol at room temperature to give the desired amine here in excellent yield on 30-gram scale. The combination of an excess of sodium borohydride and cobalt chloride in refluxing water proved to be a suitable reagent system for the efficient reduction of this azide here. It should be emphasized that under these conditions, no reduction of ketones at C3 or C20 was observed. Reduction of azides with tributyltin hydride in the presence of a radical initiator, in general AIBM, occurs via the formation of, this radical species, A or B, a 1,3-triazenyl or a 3,3-triazenyl radical, which afford staninic aminyl radicals here through the loss of nitrogen. It is worth noting that stannyl aminyl radicals here are able to perform intramolecular cyclization very efficiently with various functions, for example, alkenes, carbonyls, and nitriles. In the context of the preparation of new drug candidates to treat infections from this bromoazide here, clean debromination and reduction of this azide function here was achieved by treatment with 4 equivalents of tributyl hydride in the presence of AIBM to give, after acidification with dilute HCl, the corresponding 4 amino hydrochloride salt here in a 79% yield. Reduction of both the C4 azide function here and C3 here of this substrate using 4 equivalents of tributyl hydride in a hot dioxane in the presence of ABN as a radical initiator, followed by protection of this amine here as benzyl carbamate, afforded this derivative in excellent yield on 10-gram scale. Interestingly, as presented in this example from this iodo-azido sugar here, treatment with 1.2 equivalents of tributyl hydride in a hot toluene in the presence of ABN as a radical initiator gave chemoselectively this debrominated azido adduct here in good yield. Under the same conditions, but this time with the 5 equivalents of tributyl hydride, the debrominated amino derivative was isolated in excellent yield, as you can see. In this other example, radical mediated reduction of this 2-azidolactone here gave the corresponding 2,3-unsaturated 2-aminolactone in a good yield on 2-gram scale without any traces of debrominated compound here. So in contrast to the previous example, the azide in this case was reduced first. An alternative attempts to prepare this amine by a Staudinger reduction using triphenylphosphine to reduce the azide was not successful due to the instability of the vinylic iminophosphorane intermediate here. As previously mentioned, stanyl-aminyl radicals are able to perform intramolecular cyclization very efficiently with various functions. In this example, free radical reduction of the azide function led to this nitrogen-centered radical here, which cyclize onto this silylated enol ether Z to give the corresponding pyrroline here in good yield and with an excellent diasterose selectivity, as you can see here. Thiol reagents have also proved to be very useful in a wide range of azide reductions, as presented in this section. As pointed out by different authors, 1,3-propanediol in the presence of triethylamine is a chemoselective reagent to reduce azides. In this reduction process, 1,3-propanediol attacks the azido function to give the first intermediate EA, which, through a series of proton exchanges mediated by triethylamine, provides intermediate C. The rearrangement of this intermediate C with expulsion of nitrogen gas provides the amine with a concomitant formation of this cyclic disulfide adduct here. For example, this aromatic chiral azide treated with an excess of 1,3-propanediethiol and triethylamine in methanol gave the corresponding amino derivative in, oops, I have to move, sorry, give this compound with a 67% yield on 2-gram scale. Under the same conditions, reduction of this highly hindered azide here with 1,3-propanediethiol gave the corresponding amino derivative here in 66% yield on a gram scale. In the in the final stage of total synthesis of this natural product, reduction of this azide using a combination of 1,3-propanediethiol and triethylamine in aqueous pyridine gave the desired amine here in excellent yield, even if the scale, as you can see, is not very high. Reduction of this compound by hydrogenation was, of course, unsuitable due to the presence of several unsaturations here. Attempts to use Staudinger reduction led to poor isolated yields and various side products. In the synthesis of enantiopure alpha-aminophosphonic acids, reduction of this azide here with an excess of 1,3-propanediethiol and triethylamine in methanol gave the corresponding amino derivative in good yield on the gram scale. To avoid handling large quantities of 1,3-propanediethiol, which is an unpleasant smelling reagent, a catalytic version was developed with sodium borohydride as a co-reducer, itself inert on the azide compounds, as already mentioned. 1,3-propanediethiol is regenerated from that disulfide formed during the reducing process by reduction with sodium borohydride. This procedure was applied to this azide to give, with only 1 molar percent of 1,3-propanediethiol, the amino derivative here in almost a quantitative yield on 5-gram scale. Villarasa and collaborators have described an efficient procedure to reduce azides using a thin Mercaptan system as presented in this last section. Tin chloride, benzene thiol, and trimethylamine are mixed together to give presumably this complex, which is a reducing species. The following mechanism for the azide reduction has been proposed. Apparently, this reduction process does not involve a free radical chain mechanism. Tin-2-sulfur complex here attacks the azide function, followed by a series of proton exchanges mediated by triethylamine and expulsion of nitrogen gas at this stage to provide this intermediate here, which after hydrolysis gives the desired amino compound. This procedure was successfully applied to the reduction of this azido alcohol here to give the corresponding amino alcohol in 70% yield. Interestingly, reduction of the ketone derivative of this intermediate here with one equivalence of LH at minus 20 degrees Celsius afforded a mixture of trans here and cis azidoalcohols, while no reduction of the azide function was observed. In the total synthesis of a complex cyclic natural product, this highly functionalized azido intermediate was reduced by the Villarasas procedure to give the corresponding amino derivative here in excellent yield, as you can see. In this later example, initial attempts to reduce this azide here to the corresponding amine by Staudinger reduction or with 1,3-propane dithiol failed due to the formation of numerous byproducts. Using Villarasas procedure, the amine was obtained in a 72% yield. So, in conclusion, a range of methodologies to reduce azides to amines have been illustrated. The number and diversity of milder methods available to carry out this transformation in a chemoselective way provides an ideal opportunity to use azides as a precursors of amino compounds in various fields of total synthesis of natural products or for the preparation of pharmaceutical compounds. So, in more than 20 years of chemistry for our customers and more than 2,000 contracts completed, covering a large type of chemistry, we have used at one time or another these methodologies. Since 2004, we also ensure an important scientific watch, particularly with our scientific letters, our webinars, and with the Minute of Chemistry on the LinkedIn. We have a strong expertise in the synthesis of labeled products, more than 300 compounds prepared. We have also a strong expertise in the isolation and synthesis of APIs impurities. So to contact us, you have here the email and phone number of Aurélie and Elena, our sales manager. Thank you for your attention. I would like to thank all my colleagues from AtlanChim Pharma for the discussions during the preparation of this webinar, and Aurélie and Elena for the organization. I would like also to thank Laurence Arzel for her careful reading and her relevant remarks on the first version of this webinar. Thank you again for your attention, and now I think it's open to discussions and two equations. Okay, yes, it's interesting point. In fact, in the literature, the reaction of sodium borhydride and nickel chloride in aqueous or alcoholic solvents has been reported to give nickel boride. And nickel boride is able to promote the hydrogenation of azides with hydrogen formed um, during the reaction of sodium borhydride and methanol in our case. And you have a kind of reactivity which is very close to Raney nickel. Oh yeah, yes, it's another interesting point. In fact, I checked it in the paper why they use sodium hydroxide in this aqueous solution, but I didn't have an explanation. It's maybe because they don't want to protonate the amines at the end. I don't know, but I have no explication about this treatment because, in fact, as you saw on the other conditions, they always use acidic media, you know, or acidic source such as HCl, for example. I saw very few example with zinc borhydride and other metals. I think it's most of the time it's nickel chloride and cobalt chloride. I, but I don't have other example on this point. But maybe it's possible to have other salts. Yeah, that when I checked through the literature about reduction of azides with tin tributyl tin hydride, I saw a couple examples in which the tributyl tin hydride was using alone and without radical initiators. And maybe in this case, you have reduction by hydride transfer, like with the other hydride. And in case, in one case, I saw these conditions, and they did the reaction in toluene at room temperature. Yeah, I agree with you. It's on large scale. I don't think it's a good thing to use azides as intermediates. But in the lab, I think it's very interesting because for example, azides are very tolerant to oxidation conditions. For example, in one case, we had a compound which was, of course, a azide derivative, and it was possible, for example, to do reaction with ozone, to do ozonolysis. And I think it's interesting. I think the only um, weakness of azide is reducing conditions. But even, with sodium hydride, most of the time, azides are stable. Maybe to use acetic acid, maybe it will be a solution. I don't know exactly. I didn't have in mind example in this way. Oh, that it's a, I think, very important point. And from a point of view, I think ethanol is it's the best solvent to do hydrogenation, to do hydrogenolysis of, for example, of benzyl amines or benzyl ethers, because you have less side reaction compared to methanol. And sometimes to do catalytic hydrogenation, sometimes THF uh, could give good results as well, which is very surprising because we always think that we should have a protic solvent or at least kind of polar solvent. But sometimes THF it's a very interesting solvent. In in particular, when you want to cleave benzyl ether by hydrogenolysis, it's a very good solvent. Okay, I would like to thank you again and wish you Merry Christmas and Happy New Year. So bye bye.